My name is Cengiz Cinnioglu, and I would like to thank Thermo Fisher for having me to discuss the most recent advances in PGT-A pre-implantation genetic testing world. So this is my disclosure. I'm a founder and the CEO of NextGen Genetics. Today I would like to cover these topics. First, I would like to start PGT-A technologies, and I would like to mainly focus on ReproSeq verification. And I would like to also talk about one of the recent most hot topic about embryo mosaicism and how we are able to do this analytical testing with the ReproSeq platform. And we will also go over a little bit the cutoff values to understand how the embryo mosaicism called in the field, and then we will review a few papers related to those topics. Towards at the end of the talk, I will be mainly talking about advantages of using ReproSeq platform and compared to others that are currently available in our field. Once the PGT-A talk is done, then I would like to change subject to pre-implantation genetic testing for single gene disorders. And first, I would like to actually start with current testing platform that has been utilized in our field. And I will start with the linkage analysis with the short tandem repeats, and then continue with the carrier mapping. This is SNP-based array technology. But I would like to mainly focus on NGS-based PGT-M with PGT-A and how we verify the technology through actually Thermo Fisher platform. And at the end, again, I will also discuss the advantage of using this PGT-A over any other current platform that are utilized in the field. So again, I'd like to start with the pre-implantation genetic testing for aneuploidy and explain the most recent advances in our actual field. So I put this slide just to give you a little bit an idea about NGS platform. So there's probably a misconception in our field. There is not only one NGS platform in the field. There is actually multiple of them. Each actually technology actually come up with the brilliant idea to be able to sequence samples. Some of the actually technologies actually has a high throughput, but actually low coverage or low actually readings, versus some of the actual other technology has low throughput but has a high actually sequencing reads. So it depends on actually our needs. There are multiple actually NGS platforms that are currently in the field. But when it comes to pre-implantation genetic testing, there aren't a lot, and there are only few platforms that have the full solution, which I will discuss. ReproSeq platform is going to be one of my main topics today. So before actually I jump into the NGS technology and how the NGS works, first I would like to quickly go over how we do the testing in an embryonic level. So as we all know, when the embryo actually ready in the blastocyst stage for biopsy, the embryo has two main compartments. One of them is inner cellus, which is eventually going to be the baby, and also the trophectoderm, which is going to be eventually placenta of the baby. So what we know is that removing few cells from trophectoderm, it doesn't harm the embryo, and by getting few cells from this part, we'll be able to understand the genetic makeup of those embryos. But as we all know, each cell contains approximately 6 picogram of DNA, which if 5 or 6 cells are removed from the trophectoderm, we don't have actually enough DNA to start with to use in any platform. So in order for us to be able to actually increase the DNA yield, we actually use a technology called whole genome amplification. So once we get those few cells from biopsied embryos, we first lyse the cell, and then once the DNA is available, we actually do the amplification to be able to increase the yield thousands of times that we can use any platform for the genetic testing. So once actually DNA is available, then what we do is we actually fragment the DNA in small pieces, and they are approximately 100 to 200 base pair. And we put like small adapters that allow us to be able to start sequencing library. And those each small fragment will be actually sequenced millions of times. And at the end, we will actually get all the small pieces and compare to reference DNA, which has come from the Human Genome Project, to be able to understand copy number for each chromosome for an embryonic level. So one of the actually advantages with next generation sequencing that we did not have before with other platforms such as ArrayCGH or SNP is the barcoding. If you think about actually next generation sequencing has been around for the last 6 or 7 years. But previously we were using in the early stage of this genetic testing analysis, we used to use actually a FISH technology, fluorescent in situ hybridization. But of course, this is not molecular approach. When we move to molecular approach, we start using ArrayCGH or SNP. And I believe they did really good job in the past. The only problem with those platforms, we did not have actually barcoding system that we can actually do simultaneous analysis, multiple samples in the same reaction. With the next generation sequencing, we are actually able to barcode every single sample. And initially we were able to only mix up 24 samples and we were able to sequence them in parallel. But now we are able to actually barcode 96 samples. And after the barcoding, we can pull all of them in a single tube and we can do parallel sequencing for each sample, which reduces the cost significantly. One of the other big advantages with next generation sequencing is the higher dynamic range. Now, just because of the higher resolution, we used to actually call the embryos normal or abnormal, like as if everything is black and white, which we know that when it comes to biology, nothing is in black and white. Now, with this due to higher dynamic range, we are able to also look at the mosaic status of the embryos. Although we are calling like mosaic status, but some scientists would like to use intermediate result, or some scientists would like to use putative mosaic. So, but whatever we call them now, due to with these newer technologies, with the NGS, we are able to again not only call aneuploidy and euploidy, but also mosaic status of the embryos as well. And again, due to higher resolution, we can also identify small segmental rearrangements, which help us to be able to also do all the testing for translocation cases. And like as I mentioned, one of the important part is also the cost. And now compared to any other previous platforms such as ArrayCGH or SNP, the cost is significantly lower with next generation sequencing. With that, actually, many people will have an access to those such technologies. So, till recently, we were discussing whether or not PGT-A should be used in our field. But I think this discussion pretty much is over. Now the question is not really whether or not we should be using PGT-A technology for embryo or any other analysis. The other question is how those NGS technology can actually address those mosaic embryos, which is intermediate results where they are not normal and they are not abnormal. Now, before jump into the mosaic embryo analysis, I would like to just go over what is mosaic embryos. By definition, finding more than one distinct chromosomal count in a sample. And the reason of those mosaic embryos, it could be either due to anaphase lack or mitotic missegregation of the chromosome or premature cell division before the DNA replication. So the timing of the missegregation it also affects the severity of the mosaic embryos. And if you look at actually from the published paper, incidence of euploid, aneuploid, and mosaicism varying as low as 2% and as high as 30% in the blastocyst stage. What we know actually, even from perinatal specimen, we are finding up to 0.25% in amniotic fluid and also up to 2% in chronic villi sampling. So the mosaicism definitely is a fact. Now, we still have actually a lot of unknown about mosaic embryos. Like as I said, some scientists doesn't want to necessarily want to call those mosaic embryos as a mosaic. They want to call them intermediate result, which I would not disagree with this. Because when we call mosaic embryos, we don't even really know how many embryos are truly mosaic, how those biopsies correlate to those inner cell mass, and also how those actually, the biopsies actually affect even overall mosaic embryos. The way that we grow the embryos or handling the embryos doesn't really impact the overall mosaic results. And when we transfer those mosaic embryos and when we have a healthy babies, are they really truly normal? So with this actually today webinar, we'll kind of try to answer some of those questions that we just like listed in here. So before we talk about a day 5 embryo mosaicism, I would like to actually quickly touch base on mosaicism on day 3. Till recently, all the genetic testing effort was done on day 3 embryo development. Um, and ideally, in day 3 embryo development, you have a typically 8 cells, and just by chance, one cell may divide abnormally. And in case the biopsy is done with this purple cell, which is the aneuploid cell, and the result obviously will come abnormal. Whereas just by chance, if the euploid cell was removed, the result will come back normal. But if you look at actually this scenario, in both cases, the result is correct, but both of them doesn't represent the whole embryo just because of this embryo mosaicism. In fact, in the past, when we were doing day 3 biopsy, and then we were doing reanalysis on day 3, in some cases we were finding the abnormal result was corrected on day 5. And I don't believe the result was corrected. I don't think the embryo is going to be suddenly smart and try to correct by himself. I think what happens is that if the euploid embryo is removed, and the sister cells is the only one is going to remain in the embryo, so by removing the aneuploid cells, we're basically cleaning the embryo. And if the sister cells died out in some point, the embryo will have only good cells, which is euploid cells, cell lines. And if you ever do another biopsy in day 5 or in day 6, the result will come back normal. Now, the question is the technology is incorrect, or it's just like biology is kind of giving us those results, which unfortunately cause either false negative or false positive results. So I think we really did good job when we were doing day 3 biopsies. Not that I'm suggesting that we should go back to day 3 biopsies, but I think if you look at what's happening with day 5 biopsies, I don't think a lot of things have changed since then when it comes to embryo mosaicism. And I remember when we were doing day 3 biopsies, we were hearing actually from scientific paper, 15% of those day 3 embryos are mosaic. So we decided to actually stop doing day 3 biopsies just because of it's only a single cell, and we moved to day 5 biopsy because we believe that the mosaicism on day 5 is definitely less. But if you look at actually those scenarios, the mosaicism actually can be kind of be different for each scenario. So in some cases, trophectoderm could be affected, but not the ICM, inner cell mass. Or sometimes half of the ICM is affected, but not the trophectoderm. Or in some cases, only trophectoderm affected, but not the ICM. So unfortunately, we are still facing those embryo mosaicism even on day 5, the embryo development. And in fact, those kind of scenarios very well studied in the Kapavos and his colleague with the 2016 paper. He showed that some of the actually mosaicism can be reciprocal, some of them can be uniform. So it depends actually which part of the embryo is taken. Obviously, we were not going to touch the inner cell mass for obvious reasons, but we still actually have chance to be getting those mosaic cell lines, and which may or may not represent the whole embryo. Now, if the embryo mosaicism that has a high frequency, then the question is why we are doing even the testing in the first place. What we know actually, if the biopsy represents the whole embryo, PGT-A result is extremely accurate. In fact, a paper that was published in 2010, it's a little bit like old paper, but I am also one of the co-authors in this paper. And we actually did the study with the donated embryos where we did the analysis by removing inner cell mass from the embryo, and the leftover embryo was split in two to be able to look at those concordance rates between trophectoderms and trophectoderm and ICM concordance rate. With that specific paper, when we look at the trophectoderm biopsies, we found 100% concordance between all the trophectoderm results. But when we compared the trophectoderm result with inner cell mass, we found 96.1% concordance rate, which was quite high. And also that kind of tells us this genetic testing working in the embryonic level. But what we also find that 3.9% of the time, the trophectoderm may not present the whole inner cell mass and the rest of the embryo, which unfortunately it may cause false negative or false positive. But unfortunately, those are the reality that we have to live with. Now, a little bit actually more kind of review about embryo mosaicism. So what we know actually, when it comes to embryo mosaicism or those intermediate results, um, there are different algorithms and different cutoff values may impact the overall result. If the cutoff values is very low or high, then the mosaic embryo result will kind of differ depending on where you put the cutoff. And with that said, does PGT really help our IVF goal? And again, the same question, when we actually call an embryo is mosaic or intermediate result or protective mosaic, are they really all actually mosaic? And what is the technical contribution? And where do we go from there? Now, what we know is actually there is another research field that's already started for the last few years in non-invasive pre-implantation genetic testing for aneuploidy, which is, this is not the today topic, but based on those new research, the culture media where the embryos actually grow may have a better representation of the whole embryo rather than just removing few cells from perfected out. Now, I would like to actually go a little bit deep inside of this like embryo mosaicism call and how those actually the call was made based on different genetic labs. So it's not actually about like when we make the mosaic embryo call, this is not about mainly which NGS platform we are using. It is mainly depend of the operator and the cutoff values. In some labs, you may have the cutoff value anywhere between lower cutoff is 20 and higher cutoff is 80. And on the other hand, some other cutoff values goes to anywhere from 30 to 70. So obviously, if you go with those lower cutoff versus higher cutoff, then there is 10% like a clear difference in here. And if you are using lower cutoff, obviously you will have more mosaic embryos or more intermediate results. Again, what we know is that obviously NGS platform has definitely some kind of impact on the overall result, but the lab protocols, cutoff values, algorithm, whole genome amplification protocol, all unfortunately contribute to our embryo mosaicism call. And as far as I know, there is not really kind of really standardization in our field. And when you look at all the papers that are recently published, varying cutoffs let the percentage of the mosaic embryos reported anywhere from 5% to 20%. And if you think about 5 to 20%, there are 15% differences in the embryo mosaicism call. Now, I would like to actually quickly touch base on PGT-A's guideline. And in 2016, PGT-A actually published the guideline for transferring mosaic embryos. But if you look at their recommendation, I pretty much agree with everything that they actually recommended. And they recommend only transfer mosaic embryos if the couple's patient doesn't have an alternative embryo exist. And transferring mosaic embryos should be done only with proper genetic counseling. But the disagreement that I have, actually, this is my personal opinion, is the technical cutoff. So initial cutoff values actually was given 20% or less should be considered normal, and anytime there is elevation from over 20% all the way to 80%, that's where we'll be considering a mosaic calls or intermediate result, and anything more than 80% should be considered abnormal. And they actually give also some transfer priority should be given to mosaic monosomy for obvious reason, with a few exceptions over the trisomic embryos. But in 2019, the same PGT-A guideline actually had pretty much everything same, remained the same, except the technical cutoff values is actually changed from 20% to 40%. In my opinion, 20% was low cutoff. And we need to keep in mind that all the technologies that we are using are not perfect, especially whole genome amplification and time to time or embryo biopsy may contribute, unfortunately, those kind of like noisy actually reading. And having 20% cutoff may be confused actually with mosaicism call. But at the same time, PGT-A in 2019, actually they increased the lower cutoff values to from 20% to 40%. In my opinion, again, 20% was low, and I believe 40% is high. Part of the reason is that probably we're going to start to see more implantation failure if the couple ever transfer those mosaic embryos, and because or the implantation failure or actually implantation with some miscarriage due to the high cutoff holes on those embryos. Now, I would like to actually kind of slightly switch the subject from those mosaic embryo calls to analytical verification that we have done with by the research base with the Coriell cell lines to actually look at the ReproSeq protocol with Thermo Fisher. What we did was in our laboratory, we actually purchased Coriell cell lines. Those are commercially available. And there are plenty actually normals and abnormals, normal males and normal female cell line that can be actually used for the analytical verification. And here in this slide, you could see actually two profiles. So I would like to start the ones on the top. This is actually normal 46XX normal female that we purchased from Coriell. So with this profile, I would like to explain to you what you're seeing here. With the x-axis, you would see the number goes from chromosome 1 all the way to chromosome 22. And then we have the sex of the cell line, which is, in this case, is X and X. If you look at the y-axis, the number goes from 0 all the way to 3.5. So in order for us to call anything is normal, we need to have two copies. One copy comes from maternal side and one copy comes from paternal side. And any deviation from those numbers we would be considering abnormal. So obviously those are actually a control sample. We know what they are. We actually removed a few cells from those Coriell cell lines and pretend as if they are like single cell or 2 cells or 5 cells. And we run them on our ReproSeq protocol, and we were able to get picture perfect, nice, clear, normal male and female. If you look at the second one, actually, this one is the normal male. The only difference, obviously, between normal male and female is there is an extra Y chromosome in the male profile. And but again, all the chromosomes are listed in here. Those are picture perfect, 2 copies for every single chromosome. Now, yes, we can detect the normal and male or female. Now, the question is, how are we going to be able to detect those abnormal cell lines? Again, we also purchased Coriell cell lines, abnormal for analytical verification. And we actually had monosomy X. As you can see in here, in the circle in the first picture, we have 2 copies for every single chromosome, except there is only one X and there is no Y. We also had some chromosome where there was an addition. And in this case, we have a trisomy 13. And as you guys know, when we have an addition, those are actually indication for trisomy, and there is a missing chromosome. Those are indication for monosomy. So with the second example, the ones in the middle have 47 chromosome, and there is a trisomy 13. And instead of having 2 copies, there are actually 3 copies. Again, those are really, really nice, crystal clear, picture perfect profiles. Those are good, actually, DNA quality. And when you have actually very nice good DNA quality, those technologies pretty much always works. And the last one is actually trisomy 18, where there's an addition for chromosome 18, and it's also depicted with the blue circle at the end. Now, we are able to detect those full gain or losses, but how about those segmental errors? Are we able to detect those segmental errors? We also purchased some of the Coriell cell lines that has those segmental errors for analytical verification purpose with ReproSeq protocol. The first one, actually, as you can see, it's 46XY, and there is a duplication on chromosome 3. And this is approximately 62.2 megabase duplication. And as you can see in here, on the chromosome 3, there is no complete gain. It's just like part of the segment is duplicated. But if you look at actually the second one, we said like, what is our resolution? Like how low we can go, and we will be still able to detect those deletions or duplications. If you look at with the second example, where then we have a deletion on chromosome 18, that specific cell line had 7 megabase deletion. And as you can see in here, we can still detect those 7 megabase resolution, which is quite high, good resolution. And the last one is on the chromosome 9. Then in the P arm, there is actually duplication, and we were also able to detect with the 4 megabase duplication for segmental errors. Being able to detect those segmentals is really important because we will be able to also address some of the translocation cases that happens in the IVF world. Perfect. Now we know we can detect those full gain or losses, and we can detect the segmentals. But now the question is, how about mosaic holes? Are we going to be able to detect those? So what we did was, in order to address this mosaicism detection, we did also again analytical verification by artificially mixed corneal cell lines, because from corneal cell lines you cannot really buy mosaic cell lines. I mean, there are actually some mosaic cell lines that are currently available, but we wanted to do the control analysis where we mixed up two cell lines that we know what they are. And if you look at the first example, we had trisomy 13, XY 30%, and trisomy 18, we put 70%. And again, this is not my actually real software, but if I had the real software, if we actually zoom in every single chromosome, you would be able to see the percentage-wise whether or not they are 30% mosaic or 70% mosaicism. We also mixed another two different cell lines where we had monosomy X and normal XY. We mixed up with 50%, and you can see the gender, how they look like. They look like two copies of X and then one copy of Y, Y chromosome. And then we also mixed up 50-50 with trisomy 13 and 18. And with all the kind of like experiment that we did, we actually convinced ourselves that those mosaicism detection can be done with ReproSeq. Perfect. So one of the advantages of using ReproSeq is as follows. So the whole ReproSeq protocol takes approximately 16 hours. And we do only 3 hours hands-on, which is kind of really good, because less the human actually involvement, that means the less actually the human the error in our lab. So the 2/3 of the workflow are pretty much done with the liquid handler, which is called Ion Chef, and the sequencing device. And this is the device that you can see in the right-hand side. So this is the device that I'm just mentioning. Once we have the 3 hours cell lysis and whole genome amplification protocol is completely done, pretty much the rest of the 2/3 of the workflow will be done with liquid handler and the sequencing device to be able to complete all the analysis. Due to the high resolution, as I mentioned, we are able to actually detect all the segmental rearrangements to address all the translocation cases. But for me, one of the big advantages of using ReproSeq is, first of all, is lab user friendly. What I mean by that is that we are using 96 samples in each run, and unlike the other platform, we do not really have to measure every single DNA and normalize them, and then put them in the same actually tube to be able to do the in parallel sequencing. One of the big advantages with ReproSeq is once you do the whole genome amplification, you don't even have to measure every single DNA. You just take a liquid, which is 5-fold microliter from each sample, and you pull them in a single tube, and that's how you kind of continue with the sequencing. Which, if you think about like really measuring 96 samples, and once you get the measurement, if you have to really do the dilution based on their measurement, it takes a lot of time and effort. And probably there will be a lot of human error with this process. The other also big advantage with ReproSeq is there is a detailed chromosome analysis. And with this actually software, there's an optional actually embryo mosaicism called with the Ion Reporter software. And again, one of the most important things, as I mentioned, the less human involvement, that means less error due to this liquid handler, which is called like Ion Chef. One of the other big advantages with ReproSeq is the integration. As you can imagine, every time we sequence an embryo or any sample, we get like massive data. We get a lot of information. Obviously, we cannot really enter those like manually. So the integration for us was really crucial. And with actually Ion Reporter with Thermo Fisher, they have actually a very nice integration system that whichever database that you use, due to their integration system, you can easily actually transfer all the information automatically to your database, that you don't actually have to type or you have to actually write anything the way that we used to do in the past. So as you can see here, the ones actually on the top, this is our database. And for each embryo that we get result, they will be automatically dumped into the database that we are using. And obviously, we still check all the results against the profile that we receive just to make sure that we are not missing anything or nothing has been actually missed during those transfers. Again, I want to bring this actually video again because this is one of my favorite devices, and I just want to play the video again. This is liquid handler and called the Ion Chef. This protocol takes approximately 5 hours from the beginning all the way to the end, which takes a lot of burden from any lab. But one of the actually big advantage with the system is you can actually customize or you can program this robot based on your lab workflow. Like for instance, when the whole run is completed 5 hours later, that chip that needs to be actually transferred to the next generation sequencing platform. But if you don't have any person when the run is completed, then what you could do is actually with this Ion Chef, you can program this to overnight running, and then you can program it for following day in the morning. So the device will work all day long, all night long, and then when you come back to the lab, the chip will be ready actually to upload to next generation sequencing device. Another good thing about with those devices, as long as you kind of do the maintenance, those device doesn't have a good days and bad days, unlike the human being. And just because of those devices, the human error rate will be significantly reduced or minimized. Okay, so after all this repressive information, I would like to review a couple papers that I would like to go over in terms of the concordance rate between your cell mass and trifectodome, and concordance rate between mosaic embryo call and the mosaic embryo transfer and some of the outcomes. So this is one of the most recent papers. Like as I mentioned earlier, although we did not have a lot of data point at the beginning, now with this recent few papers, now we have actually more information about those cutoff values in the mosaic embryo transfer. And I would like to start with the paper that was published with Dr. Viotti and in 2020 in his colleague. They use the cutoff values about 20 to 80%. Like as I mentioned, 20% could be a little bit low end, but that's what the paper actually mentioned. The cutoff is anywhere between 20 and 80. And they were able to also categorize those embryos low mosaic or high mosaic. Low mosaic means if the cells has like less than 50% or more than 40%, those aneuploid samples, aneuploid actually cells in the embryo, and that's how they were categorized. If you look at the overall results, and the euploid embryo has a better better outcome compared to combined mosaic embryos or single chromosome mosaicism, which is kind of expected. When you look at the spontaneous abortion, with the euploid embryos, it's at about 8%, but with the any mosaic embryo transfer, the outcome was higher when it comes to spontaneous abortion, which is 20.4% and 25%. And in this paper, they categorize the embryos in two sides. One is lower mosaic, where we assume that lower mosaicism, meaning that we have more healthy cells compared to abnormal cells. And if the embryo has actually more than 50% mosaicism, that means we have more abnormal cells and less actually healthy cells. And when those transfer was done, lower mosaicism versus high mosaicism, the implantation actually significantly differed between the lower mosaic embryos had a higher implantation and the ongoing pregnancy compared to high mosaic embryos. The assumption again with the lower mosaicism, we have like more healthy cells compared to abnormal cells. But one thing that I also want to emphasize or actually mention is that if you have a low cutoff values like 20, of course I'm speculating, don't know the answers. Some of those low mosaic embryos may not be even actually mosaic in the first place, and that was the reason maybe we are getting some successful result from those low mosaic embryos compared to higher mosaic ones. And in the same paper, they looked at the embryo mosaicism type, whether or not low, high, whether or not they are complete chromosome gain or losses, or versus they are segmental mosaicism. And what they found specifically with this paper, segmental mosaic actually had the better outcome compared to any other actually full chromosome gain or losses. But overall, combining with the mosaic level, whether or not high or low, and the type of the mosaicism, again, whether or not this is full chromosome gain or losses, or segmental, and with the combination of embryo morphology, significantly actually affected the likelihood of the positive outcome. This is another actually not necessarily paper, a small abstract. I really wanted to put this just to kind of to show you when you use lower cutoff, 20 versus 30, and the upper cutoff, 70 versus 80, just because of depends on where you put those cutoff values. Depending those cutoff values, the percentage of the mosaicism embryos reported anywhere from 4.9% to 17%, which there is actually 12% difference in between two numbers. So with this paper, the assumption is it's not about like which next generation technology you are utilizing. It is the most important thing is where the cutoff will be placed. And due to high cutoff values, and the one genetic lab may have a higher euploidy compared to other genetic labs for obvious reasons, depends on where you put those thresholds. This is another interesting paper that also show the value of genetic testing, PGT-A. Time to time, we get requests for rebiopsy embryos. When you look at the specific paper, they look at the concordance between the first and second biopsy in mosaic embryo results. When they look at actually those rebiopsy on euploid embryos, the concordance rate was quite high, 9.5%, which is really amazing. And when they look at the aneuploid result, they were also really quite high, which is 97.3%. But if you ever do rebiopsy on mosaic embryos, the concordance rate is quite low, which is about 35.2%. If the first biopsy comes mosaic chromosome 5, and if you were to do another biopsy, you may get the same result, or you may get like another maybe mosaic chromosome. But that's the reason we are calling those embryos mosaic. And that's why every time you do another biopsy, probably you may end up with a different percentage. But that's why we call them mosaic. And it's not about which result we should trust. All the results is correct. It's just the embryo by itself is mosaic. So after talking a lot about pre-implantation genetic testing for aneuploidy, I would like to talk about pre-implantation genetic testing for single gene disorders. And this is going to be my next topic and the last topic. So I would like to actually quickly go overview about what has been currently done for single gene disorders in our field. So all the initial effort for single gene disorder is mainly done either with linkage analysis or direct mutation analysis, and actually often is combination of both. But all the initial effort for the single gene disorder was done with the short tandem repeat STR analysis. So as we know, the genome has exon, which is the coding region, and intron, which is non-coding region. And there are a lot of actually STR are located in the intron region. They are just keep repeating. Some of the actually repeat can be 4 base pairs. It can be like ACTA, keep repeating a few times. We don't know why they are even exist in the intron, but we have been using those for all different kind of purposes. STR, short tandem repeat, has been widely used in fluorescence application. But we are using STR analysis for linkage analysis in case we want to do direct mutation analysis. It's just because of the DNA that we are working with require whole genome amplification. And unfortunately, whole genome amplification, there are some limitations. Some part of the genome may not work. And the part that we are interested in, if it doesn't amplify, then we have to be actually using linkage analysis to understand which homolog chromosome goes from parent to their embryonic level. Recently, single nucleotide polymorphism has been introduced to the R field, and this is mainly Illumina Infinium 2 SNP array. This is called like array-based linkage by karyomapping. With this technology, this specific array consists of 300,000 SNP, and to be able to do the linkage analysis. So single nucleotide polymorphism actually more common than the SDRs in the genome, and it's estimated 4 to 5 million SNPs can be found throughout the whole genome that can be utilized for forensic application or any linkage analysis. And the last one that I want to discuss, which is the technology that I'm currently using through Thermo Fisher, it's called PGT-A, and this is still SNP-based approach, but instead of using array platform, we're going to be using next generation sequencing for linkage analysis. So I would like to start with linkage analysis with STR. Like as I mentioned before, the STR analysis basically where there are some repeats, and every human being carry 2 chromosomes, one comes from mom, one comes from your dad. And with that specific example, let's assume that mom has ACC repeats. So this is only one STR marker that I would like to give you an example. And as you can see, one of the allele with the mom has 5 repeats. ACC is repeating 5 times. The second allele for the same mom has, in the second chromosome, has 7 repeats. If you look at that, that has 6 and 7 repeats. And again, this is only one STR marker that I'm talking about. So now the mom has heterozygous 5-7, and the dad is heterozygous 6 and 7 repeats. So the possible alleles in the embryonic level would be either 5 to 6, 7, 5 to 7, 6 to 7, or 7 to 7 repeats. Those are the possible alleles in the embryonic level. So ideally, when we are going to do single gene disorder, we would like to sequence the disease mutation, and that will tell us what's going on in an embryonic level. Just because of we are using whole genome amplification, and just by chance, if that specific disease region that we're interested in is not amplifying, then we cannot actually give the answer about the embryonic level. And that's why we use linkage analysis with the STR testing to be able to understand which homolog chromosome coming from maternal side to the embryonic level and which homolog chromosome coming from that to the embryonic level. And based on using those the STR markers, we will be able to understand the embryonic level and whether or not the embryo is affected or carriers or completely healthy. When it comes to STR analysis, they are typically we typically develop 6 or 7 STR markers surrounding the disease mutation. So we try to be as close as possible to the disease mutation. And one of the reasons is to make sure that the crossover doesn't interfere with our resulting that cause possible misdiagnosis. Typically, 6 or 7 STR is usually utilized for linkage analysis, and in some cases, combination of direct mutation analysis as well. Now, the second approach is linkage analysis with single nucleotide polymorphism for monogenic disorders, and one of the technology that has been widely used is carrier mapping. With this technology, instead of having repeats, there are actually single nucleotide substitution, and there are approximately 4 to 5 million SNPs that can be utilized throughout the whole genome. And in this case, the mom is assumed as a GG allele and the dad has TT allele. And if you put them in the context of homozygous AA or homozygous BB, and you would be able to kind of find the possible alleles in the embryonic level, whether or not they are AA, BB, or AB alleles in the context of this parent information. This again applies the same thing, like ideally we would like to sequence the disease mutation if we can, but in case we cannot really have really nice reading in that specific disease mutation, then we would be using those SNP markers. SNP markers actually has more frequency. Actually, they are more common in the genome. Again, compared to thousands of SDRs, we have actually over 4 or 5 million SNPs throughout the whole genome. So, as I mentioned in my previous slide, when we design STR markers, we try to be as close as possible to disease mutation. But with the STR analysis, in some cases, we might not get lucky and we may not be able to find those STR markers close by. But with the SNP, they're actually more powerful because there are like plenty of them to use in the genome. And with this technology, with the carrier mapping, we are actually able to find close to 30 single nucleotide polymorphism flanking region of the disease mutation. Obviously, it's better than having only 5 or 6 STR markers, But in case of allele dropout, in case of we are not able to amplify some of the DNA that we are trying to actually search for, half of the SNP markers may not work, which would end up like getting only 15 or 10 SNP markers to work with, which is still not bad. But recently, actually before I actually jump into recent NGS technology for SNP analysis, I would like to just quickly a little bit go over carrier mapping. With the carrier mapping, you have array, and this array, that's a specific type of array, has a little bit over 300,000 SNP markers. But obviously, we cannot use all the 300,000 markers because with this example, let's assume that mom and dad has the same condition, and let's assume that this is a cystic fibrosis, where cystic fibrosis is autosomal recessive condition where the mom and dad has the same mutation. And if you think everything is color coded in here, so let's assume that the chromosome actually blue has affected chromosome, and the mom, the green one, has actually affected chromosome, which is depicted with the star. So depend on where the diamond or the star goes, the embryo could be completely affected, or could be carrier like their parents, or could be completely healthy. So it depends on where those stars or diamond goes, we'll be able to make the analysis in the embryonic level. But again, the most important part is when we look at those SNP markers, we need to be able to actually be as close as possible to the disease mutation. We cannot really kind of like look at those SNP markers that are completely unrelated to chromosome. One of the disadvantages with the carrier mapping is you are limited to whatever is deposited in the array. So if you have any mutation, when you look at the specific coverage for that specific mutation, if you don't have enough SNP markers, then you might actually have to use STR analysis to be able to kind of do the analysis. So if there is not enough coverage for that specific mutation. Now, this is the one that I would like to discuss today, linkage analysis with single nucleotide polymorphism for monogenic disease order with PGT-C. It is pretty much identical to SNP with array. The only difference is instead of using array, we're going to be using next generation sequencing. Why is important? Again, everything is applied to what I just explained previously. We try to do direct mutation analysis, but in many cases we'll probably fail, or we're not going to get really crystal clear answer. So analyzing actually those SNP markers would be really important. And one of the advantages by using a PGT-C is we can develop a panel, and we can put as many as SNP markers that we can put next to the disease mutation. And we typically have 200 SNP markers compared to only 30 or 40 markers with array-based technology. And this is how the workflow works, how the lab protocol works. When we actually do the analysis, we use Coriell cell lines. And by analyzing the cell lines for pre-implantation genetic testing for aneuploidy, as soon as we do the cell lysis and extracting the DNA, in the second step, which is right after the pre-amplification, we can take 3 macroliter aliquot, and we can continue in parallel for PGT-6 for single gene disorder. And by the time we are done PGT-A and PGT-M, we will be able to do simultaneous analysis from the same actually cell line to be able to get the final answer for both. And we also wanted to understand what if actually we have a sample with Coriell cell lines that we did the verification, but the whole genome amplification already completed. Now, is there any way we can go back and we did a single gene disorder? The answer is yes. If the sample has been already done for aneuploidy screening, we can always go back from the third step, which is where we have the amplified library. All the barcodes were already actually incorporated into the sample. If you take 20 macroliter aliquot, by purifying the sample, by removing all the barcodes and primer dimers from the whole genome amplification, we will be able to actually continue with pre-implantation genetic testing with the PGT-C, and we can still actually do the analysis. which we did. And this is how the analysis works. And this is, again, one of the samples that we received from Corial cell lines to be able to understand how the linkage works. This specific example where autosomal dominant disease, the male affected, female came negative from that specific cell line. And we had also maternal DNA from the male, which the inheritance actually came from a maternal side. So everything is color coded. As you can see, female has completely grayed out and doesn't have the mutation. Male, the blue one, the ones that doesn't carry the mutation, and the orange one, the ones that carry the mutation. So when we did the analysis, and if you look at those the Corial cell line samples, what we found is the first three samples that had gotten one chromosome from maternal side, which is definitely negative, and depend on whether or not we get the blue or the orange, we will be able to make the calls whether or not they are carrier or actually affected. The first three samples, as you can see, by chance, the unaffected chromosome was received from paternal side, but the other three samples, they all get actually the affected chromosome from a male partner with this specific Corial cell lines, which they are affected. So there's very nice software that kind of tells you what is going on for each SNP. And this is just like a small screenshot. We typically screen 200 markers, but this is the screenshot, just only show you a few of them. And even if some of the SNP that doesn't work, when you work with 200 markers, even if half of them doesn't work, we still have like 100 markers to work with. So, one of the advantages using this NGS-based PGT-M, first of all, the testing can be done on the resource basis or any basis, can be done with any mutation with the few exceptions. The testing also can be done with the small deletions or insertions if there is a family pattern. And we could do simultaneous analysis for PGT-A and PGT-M from the same specimen. And this is the list that we are able to do for any kind of genetic testing mutation analysis, but we are not limited to anything that is listed in this disease mutation list. Anytime there is any rare event mutation, we can develop the panel and we can actually target 200 or a little bit over single nucleotide markers. And this is unlike with the array-based technology. If the disease mutation that we are looking in the array platform, if there is not enough SNP markers, then that requires to use STR analysis to be able to actually do the testing. When it comes to next generation sequencing, we are not actually dependent on which chromosome and which actually rare mutation. We can develop and we can actually target those SNP markers based on the mutation that we are receiving from any specimen. The turnaround time is typically 4 to 4 weeks to develop those panels. But one of the, again, advantage using PGT-seq for monogenic disorders is that unlike the other technologies, we are using 200 markers over 30 markers with carrier mapping or 6 markers with STR linkage analysis. But more importantly, there are some common diseases like such as cystic fibrosis, spinal muscular atrophy, Fragile X, osteogenesis imperfecta, and alpha thalassemia. Those diseases are quite common in the population, and there's pretty much like no waiting period for those testing. So those actually already ready in our laboratories. And if we want to do any testing in any sample, we could easily actually perform without even waiting period, as long as there is informative family members that can be utilized for linkage analysis. But of course, if there is diseases are not covered in this 5 disease, we can definitely develop any panel, and that rare mutation panel development takes anywhere between 4 to 6 weeks. And simultaneous analysis can be done with PGT-A and PGT-M from the same sample. And this is going to be my last slide. we'll continue with using advantage of PGT-SIG. We have those ready-to-use panels, and if the couple has any of those 5 disease or any, the testing can be done easily for any research base or any kind of analysis. And the one of the also biggest advantage with PGT-SIG technology, in order to develop those panels, we are not depending on the family members for the prop development. So the prop development can start independently. Again, unlike STR, in order to actually develop STR panel, the family members having the family members first arrive to the lab is very crucial, which takes some time to collect those family members to be able to actually do the linkage analysis. But PGT-SIG with the panel development can start as soon as we would like to start, and the probe development will take anywhere from 4 to 6 weeks. And the hands-on time with the PGT-SIG takes only 4 hours, and just because of we have a liquid handler with Ion Chef, and we will be able to do majority of the work without having actually people hands-on, and most of the work will be done through the liquid handler and also sequencing device, which is S5. And obviously less human involvement and less error. And I would like to thank everyone again who has attended to this meeting. And if there's any question, I'm more than happy to take one.
Well, thank you, Dr. Cinnioglu, for your informative presentation. We will now start the live Q&A portion of the webinar. Our first question is, can DNA from amniotic sac be used as an amniocentesis rather than the DNA from the trophoectoderm of a blastocyst?
The short answer is yes. Yeah, any DNA material can be utilized for either single gene disorders or pre-implanted genetics for aneuploidy. Currently, we did analytical verification with chorial cell lines, and as long as the sample comes from amniotic or CVS chronic villi sampling, yes, we can definitely work on those samples as well.
Is this performed for checking some abnormalities in an embryo if the parent wishes?
Yes, for pre-implantation genetic testing for aneuploidy, yes, we do this testing, all the time. Yes, as long as the parent wishes, and pretty much like all the requests can be accommodated easily.
Okay, thank you. And Nisa, if you need further clarification, we'll just type that in again, and if we don't have time today, we'll do that by email. The next question is, okay, if an individual performs PGT-A but later finds out one of the partners has PGT-SR, do we need to do another biopsy, or could existing DNA be used for PGT-SR testing?
That's really great question. Yes, there is no need for the second biopsy. The DNA sample that utilized for PGT-A can be utilized for PGT-SR, and there is no really need for rebiopsy. And we just need to do actually a higher resolution, which again doesn't really require additional sampling from the same embryo, which is obviously not ideal to biopsy embryo twice. Again, the short answer is yes, and can be done SR without any need for additional sampling from the same embryo.
Okay, we've got time for one more question, Dr. Cinnioglu. I'm not sure how to say this. Is the lib prep manual or also automated?
Pretty much everything that I discussed today in here is we still use liquid handler, which is Ion Chef. So the hands-on time, regardless of PGT-A or PGT-M, the hands-on time approximately 3 to 4 hours. The whole protocol takes approximately 16 hours or 17 hours, depends on how many samples we are running. But in terms of the liquid handler, or in terms of like automation, it's pretty much the same. So we just only do a few hours hands-on, and the rest is always automatic with the liquid handlers devices.
All right. Well, thank you so much, Dr. Cinnioglu. Do you have any final comments for the audience today?
Yeah, those technologies is relatively new, And in like every other year or every year, although I've been in this field for a long time, every year we learn new things from either for any research basis or any for published paper. So I have been actually in this field for the last 15 years, but again, even it makes me dizzy kind of to see all those kind of recent advances happening in our field, which I'm really pleased to see those.
Well, thank you again, Dr. Cinnioglu, for your time today and for your important research. We would also like to thank LabRoots and our sponsors, Thermo Fisher Scientific, for underwriting today's educational webcast.